Hello, welcome to Lesson 8. This lesson is going to be on neurotransmitters. So what we're going to do is we're going to start out talking about the different molecules or neurotransmitters that are used to signal in neurons in the nervous system. We're going to be talking about different types of pathways, so we call neural pathways or different neurotransmitter systems. And we're going to be talking about different types of receptors, how they're activated, and some pharmacology. So how do we actually affect the amount and the type of neurotransmission that's taking place in the central nervous system and the peripheral nervous system for that matter. But here we're going to be focusing more on the central aspect. So those are all the things we're looking forward to in this block, and we're going to start out in this lesson just talking about the neurotransmitters. So here on the right side you see this nice image. This is a synapse. Okay, so we introduced this previously, and what you can see is two cells. We have what we call the presynaptic cell. So this is the cell where the neurotransmitter will be released from, and we have the postsynaptic cell, which is where the neurotransmitter is actually going to bind to. So neurotransmitters, we're talking about the actual chemicals, the molecules that actually are released from neurons and bind to receptors on other neurons. So this is the way that cells use chemicals to communicate. So again, we're talking about release from the presynaptic terminal. So these are going to be those terminal endings, okay? So you have your cell body here, and then you're going to have that action potential. Remember, we learned about the action potential. That's the electrical stimulation. It's going to go through the axon to the terminal, and here is where you're going to have the release of your neurotransmitter. And now on the cell body, or on these dendrites of the postsynaptic cells, where you're going to have your receptors. Okay. Now I do point out the fact that it's not always this way. It's very complex, the way that neurons signal and the way that neurotransmitters bind to receptors. So oftentimes there's actually presynaptic receptors, there's axonal receptors, so they're not always on the dendrites of the postsynaptic cell. But for simplicity's sake, we're going to just consider this situation and just say, okay, we have a presynaptic cell with a terminal. It's going to release transmitter. It's going to bind to a receptor on a postsynaptic neuron, probably somewhere in the dendrites. Okay, this is what we call ligand receptor binding. So what is a ligand? Basically, a ligand is any molecule that binds to a receptor. It's sort of a general, all-encompassing generic term for something that binds to a receptor. Now, when a ligand, such as a neurotransmitter, binds to a receptor, this is going to cause some kind of a change in the postsynaptic cell. And it's going to depend on exactly what type of neurotransmitter binds and what type of receptor it binds to. And those are more specific nuances that we're going to get to a little bit later. Now, I've included this link here because this is a nice animated explanation of how neurotransmission actually occurs. I like this one, so you can check it out if you want to. If you feel like you've already got it and it makes sense and you want to move on, that's fine too. All right, so if you watch the video, all right, then you've already seen a lot of this at play. Okay, if you didn't watch the video, then you may want to go back and watch it. Again, I'm not going to require that you watch it, but I would strongly suggest it because it's going to help you. And it's going to help you understand these different facets of how neurotransmitters occur, or how neurotransmitters signal, rather, excuse me. So the big four we're going to talk about is the synthesis. So that synthesis is to make something, right? So how neurotransmitters are actually made, how and where they're stored, how they're released, and something that we call reuptake. And I'm also going to add degradation to this too, right? But that sort of is paired in some ways to reuptake, so it's sort of like reuptake / degradation. We'll talk about that in a minute, all right? So neurotransmitters are synthesized, okay, or they are produced in the cell body of the neuron, okay? And they are housed in sacs, what we call vesicles. So you probably remember from biology class that you have these vesicles that have contents in them, right? And depending on the type of vesicle, you might have different types of things in them. Well, neurotransmitters are stored in vesicles. And the vesicles are transported, okay, through axonal transport down the axon into the terminals. And the terminals is the site where the neurotransmitters are going to be released. So in order for neurotransmitters to get made, though, they always start out from some very, very important precursors, and biosynthetic enzymes are necessary to actually catalyze the conversion of these precursors into the actual neurotransmitter. And some of these actually important precursors are necessary to make several different types of neurotransmitters, and we're going to see that in a minute as well. All right. So again, the storage, they're packaged in vesicles in the presynaptic terminal. They are released in response to an action potential. So you remember, the neuron is excited. It reaches the threshold. Okay, the cell depolarizes. An action potential occurs, which sends an electrical signal down the axon. When that signal gets to the terminals, we have the release through what we call exocytosis. So again, this should sort of be kind of review for you, at least the exocytosis portion. Now, these vesicles, okay, that remember they're synthesized in the cell body, but ultimately they are trafficked, okay, or transported down the axon into the terminal where they wait. After an action potential happens, these vesicles are either present in what they call active zones, which are very close to the presynaptic membrane of the terminal, very close to where they have to go to get out, okay? Or they're actually shuttled to the membrane for those vesicles that are a little bit further away. And this is a calcium-dependent process, all right? Calcium dependent. So the release of neurotransmitters through exocytosis is highly dependent on calcium influx, okay? So we have the influx of sodium as a major neurotransmitter for generating an action potential, while calcium is very important for actually triggering the release of the neurotransmitter out of the presynaptic cell into the synaptic cleft, all right? Now, what happens to the neurotransmitters once they bind the receptor? Well, as you know, with all molecular events, things are always binding and releasing, right? It's not like you have a molecule that's going to bind to a receptor and stay there forever. It just doesn't happen that way, okay? Molecules and binding interactions are very dynamic. And so after some time, some of the neurotransmitters will bind to the receptors and then they'll come off. They'll be released. Some of them never bind to the receptors. And sometimes you might have a lot of neurotransmitter, but you don't have much receptor there. So the point being, you're going to end up with a bunch of extra neurotransmitter floating around in the synapse. And we don't want it hanging around too long. So the cell has devised a couple of different methods by which to terminate the actions of these neurotransmitters and recycle the contents. Because we're cells, we like to be efficient. We don't want to waste anything. We don't want to waste energy, and we don't want to waste goods, right? So what it's going to do is either perform a process called reuptake, okay, where the neurotransmitter is actually transported back into the presynaptic neuron, okay, through a specialized transporter on the membrane, okay, or the neurotransmitter is going to be degraded by specific types of enzymes, all right? So either neurotransmitters get reuptaken back into the presynaptic terminal or they get degraded by enzymes. Now, what exactly are these neurotransmitters? What are these different molecules that are involved in transmission? Well, we have a few major categories, okay? And we're going to talk just for a minute about them. Essentially, any neurotransmitter, I don't care which you are, most of you, I should say most neurotransmitters, fit into a few major categories. You have your amino acid neurotransmitters, right? So these are things like GABA and glutamate. GABA and glutamate are two of the big ones in the brain. Do lots of different stuff. Then you have your amines. Now, sometimes this is separated into a monoamine or a catecholamine, okay, but they're all amines. It's just different ways of categorizing them, okay? And if you remember from your structure of an amino acid, okay, one of the reasons why it's called an amino, it has an amino group, right? It's an NH2. So amines are named amines because they have NH2 groups, okay? They have amino groups on them. All right, so they're derived from amino acids, but they have some extra things added to them, okay, and they have amino groups. So these are things like dopamine, serotonin, norepinephrine, epinephrine, and histamine. And don't worry, we're going to get into more details about these. Then we have a group called the peptides. Very interesting, very diverse group. And as you know from, again, going back to biology here, is that proteins are made from a series of amino acids that are linked together. And they're linked together through, yes, that's right, peptide bonds. Peptide bonds, okay? So the peptide neurotransmitters are a little bit bigger, okay? And there are many amino acids linked together. I'm going to show you a little schematic that shows this. So these are the three big groups. There are some exceptions. For example, acetylcholine, or ACH for short. Acetylcholine is a super, super important neurotransmitter in the body, both in the central nervous system and the peripheral nervous system. Now, this one is actually derived from choline, okay? Choline and acetyl-CoA. So if you remember anything about cellular respiration, you have production of acetyl-CoA and choline combined to form acetylcholine. All right, so that doesn't really fit neatly into one of these categories. There's also a lot of other interesting molecules that work as neurotransmitters. For example, small gas called nitric oxide, okay, NO. This actually works as a neurotransmitter. ATP, yes, adenosine triphosphate. It's the same guy, okay? The energy currency also functions as a neurotransmitter and can bind to receptors to cause different events to occur in all over the place, okay? And this is actually pretty neat because a lot of these not really acetylcholine, but things like NO, ATP, and even some of the peptides, oftentimes they're co-released with other neurotransmitters, okay? So sometimes these things work in conjunction with each other. These are different types. Now, if we look at this here, this is kind of just to show a couple of important things. First of all, this is acetylcholine here. Okay, now this, again, we said this was sort of an exception, so it's kind of in its own little group here. Here we have what we call biogenic amines. Okay, you don't have to worry too much about this word. Don't let it confuse you. Okay, these are the amines. Again, we got NH2, 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 so that sort of makes sense, right? They're amines. Now we have norepinephrine, or norepi, which I tend to call it for short, dopamine, and serotonin. There's also epinephrine. It's very similar. It's actually derived from norepinephrine. Okay, it's not shown in this chart here, but that one would also fall into this category. Now, when we talk about catecholamines would be norepinephrine and epinephrine as well, which isn't here, but again, it's very similar, and dopamine. Okay, serotonin is what we call a monoamine. Well, what's the difference? Well, a catecholamine, it's a catecholamine because it has a catechol group, which is actually this group here with these two hydroxyls on it. Okay, so you notice serotonin doesn't have the two OHs here. Okay, so structurally it's a little bit different. So that's the only reason why serotonin isn't called a catecholamine. Okay, but they are all amines. So we're just going to keep it simple. We're going to talk about amines. Just in case you see the word catecholamine or it comes up or I say it or something, don't get completely confused, all right? We're basically still talking about very similar types of ones here. Then we go to our amino acids. We have GABA, glycine, glutamate here, okay? We see a bunch of atoms, right? All these C's and H's and COH, all these carboxyl, good stuff here. Awesome bio stuff. And then we go to our peptide group. And what do we see here? Well, instead of all these neat little atoms lined up here in these sort of small molecules, we have strings of, wait for it, amino acids, okay? So these are truly peptides, right? Because these things are linked by peptide bonds. So this is the reason why these are the neuropeptides. So if you understand a little bit about the chemistry and the biology, this kind of makes sense. Now, what is not readily apparent to you yet, and what doesn't make sense, but it will, is that you see this functional class column here says excitatory or inhibitory, or generally excitatory, okay? Now, what that means is that each one of these particular types of neurotransmitters has a propensity to either inhibit or excite the cell that it binds to, okay? And really what that does, okay, the ability of a particular neurotransmitter to excite or inhibit a cell or a system really depends on the type of receptors that it binds to and what it actually does, okay? That's not going to make sense until we get into the receptor types, but for example, acetylcholine can be excitatory or inhibitory. And the reason for that is because there's different types of receptors that it can bind to. Not every acetylcholine receptor is made the same, okay? They're not all equal. And so one type of receptor will allow positively charged ions to pass into a cell, which will excite it. It'll drive it towards its threshold for firing, okay? Another type of acetylcholine receptor will actually cause it to inhibit its likelihood to fire. And it just depends on which type of receptor the cell has. So the cells have an enormous amount of variation in terms of not only the types of neurotransmitters that can bind, but also the types of receptors that they can bind to, and these really alter function. Now, when we're talking about the brain in big terms, we have two huge neurotransmitters, in GABA and glutamate. Now, these are the major inhibitory and excitatory neurotransmitters of the brain. Okay, they are all over the place. The receptors are all over the place. Okay, and in general, the brain is driven in large part by glutamate and GABA. Okay, but these other neurotransmitters will what we call modulate signals, and they fine-tune things. Okay, so they're just as important. Doesn't mean GABA and glutamate are more important than the other ones. Okay, but they are the major inhibitory and excitatory neurotransmitters of the brain, respectively here. GABA being the inhibitory, glutamate being the excitatory. And what you'll find out later is the type of receptors, okay, and the type of channel that GABA activates actually is one that lets in negative ions. So this is going to tend to hyperpolarize the cell, and that's the reason why it's inhibitory. But don't worry, we'll get to all that later. All right. Now, neurotransmitters are built from precursors. All right. So this is what we call neurotransmitter biosynthetic pathways, all right? So don't worry, you're not gonna have to memorize all these things, okay? But what I did want to show you is just an example. So here we have what we call our catecholamines, okay? Remember, these are the amine groups. So we have dopamine, we have norepinephrine, and we have epinephrine. And what you can see here is that we start off with tyrosine, right? And really phenylalanine further upstream. That's not shown here. But you have tyrosine, which is super important, right? Amino acid that we're going to begin with. And then through the actions of these different enzymes, we're actually going to do some different things to keep building different molecules, okay? So here we have dopa, or sometimes what people call L-dopa. So this is actually the precursor to dopamine. This enzyme, aromatic amino acid decarboxylase, will come in. It's a decarboxylase because it's going to remove a carboxyl group. So you can see CO2 is leaving here, all right? And that converts it into dopamine, all right? And so this is how this works, right? And all these pathways have similar functions. So here you can see tryptophan. This is one of those essential amino acids, right? One of the ones that we don't make, but we have to actually get it through our diet, okay? It's super important. This is the precursor for the production of serotonin. All right, and we actually have tryptophan hydroxylase, so this is going to add a hydroxyl group, and that's what's going to convert tryptophan through a couple other steps, where we're going to end up with serotonin. All right. Now, what's important to remember is just like in chemistry, okay, there are rate-limiting steps. Okay, so a lot of times we talk about the importance of rate-limiting enzymes in neurotransmitter biosynthetic pathways. Whew! Sounds like a mouthful, right? It's not. Sounds important. It's not that complicated. What it means is that we're building a neurotransmitter through a series of chemical reactions, right? That's a biosynthetic pathway, okay? And we have a rate-limiting enzyme, which means that the conversion of tyrosine ultimately to either dopamine or norepi or epinephrine is going to depend largely on this tyrosine hydroxylase, okay? So tyrosine hydroxylase, or TH, is the rate-limiting enzyme in, for example, dopamine biosynthesis, okay? So just wanted to go through some of that. You're not going to have to know all these structures or memorize all these pathways, but it's good to know what the rate-limiting enzyme is and what it does, okay? Now, what do each one of these things do? We're going to go through four of the big neurotransmitters, and we're going to talk a little bit about how they actually function. And we're going to start with dopamine, because dopamine is super interesting, and it does a lot of cool stuff. So, we have dopamine here you can see that we're starting with our tyrosine. We make the precursor L-DOPA, or DOPA as they just call it here, and then it becomes dopamine. It gets stored in vesicles, all right? So you're going to see similar sorts of synapses for each one of these. This is kind of just to keep you familiar with what's actually happening, right? It's not that you're going to have to know or label all the parts of this thing, but it's just we store them in vesicles, okay? And then they're going to get released into the space. They're going to bind to a receptor. Good? Good. We understand that part. Now, dopamine, the reason why it's so cool and the reason why it's so important is because it regulates lots of cool and important stuff like feeling pleasurable, okay? Anytime that you gain satisfaction from something, there's dopamine being released, okay? Reward and reinforcement. So anything that motivates you, okay, when we're talking about motivating behavior, right, and reinforcing both good behavior and bad behavior, actually, right? We're talking about, and I'm going to show you a funny clip from Big Bang Theory that I like when we get into neural systems for dopamine. But so that's what dopamine is important for. Okay, again, it has the precursor is tyrosine. So these bold-faced things are things that you guys should basically know about each one of these different neurotransmitters. So remember we said there's two mechanisms for getting rid of a neurotransmitter after it's released and bound to the receptors. One is through reuptake, okay, and the other is by degrading it. So reuptake for dopamine is achieved by the dopamine transporter, okay, and this is known as DAT. Okay, we like to keep things simple because it's lots of words and they get to be very long, and it's lots of long words and nobody wants to say all those long words, right? So DAT is a good one for dopamine transporter. So here you can see it's membrane bound, and this will actually import dopamine back into the cell. If that doesn't happen, it gets degraded, okay? Now there's two different enzymes that do this, and this is actually not only for dopamine, but for all of the monoamines, okay? One is MAO, and the other is COMT. Now, these are both enzymes, okay? This is monoamine oxidase, okay? So it's an oxidation reaction that degrades dopamine, and this is catecholomethyltransferase, right? Now that's not really that important what it actually stands for because we're not going to get into the detailed chemistry of it, but it is a transferase, it's an oxidase here, so these are enzymes, okay? And these will degrade dopamine. So it's a monoamine, so it's going to degrade it, right? D1 through D5 are the different receptors for dopamine. Now, some of these different neurotransmitters are going to have more than one type of receptor. So this is something else that we're going to be talking about in a little bit more detail later. Okay. For now, we just want to take note that there's several different types. We got 5 different dopamine receptors. All right. Lots of pathways, and we're going to have some fun with that when we get there. So what about serotonin? You're going to see this written as 5-HT. Okay, this is the standard sort of shorthand for serotonin based on its structure. Okay, serotonin, if you don't know already, super popular. It's one of the ones you'll hear about sometimes in the news. You'll hear about it on television because it is intimately connected with emotional states, as well as cognition, and it does actually is one of the important signals in the brain that controls circadian rhythm and helps you with sleep and wakefulness cycles and things like that. Okay, but it's most commonly and most commonly known based on its connections with things like depression. Okay, so you might have seen commercials for drugs, antidepressant drugs that are SSRIs. And we're going to get into this later in the course, but that's selective serotonin reuptake inhibitor. Okay, wow, that's a mouthful, right? Don't worry about it. Basically, all it means is that it blocks the reuptake of serotonin. So they're trying to keep serotonin in the synapse longer. All right. And one of the reasons is because people who are depressed tend to have lower levels of serotonin, or the receptors don't work as well, et cetera, et cetera. And we'll get into that later, okay? But this is one of those important ones. Where does it come from? It comes from tryptophan. That is the precursor. How does it get degraded or reuptaken? Well, we have the CERT for this one. Okay, so it's the serotonin reuptake transporter. So this is actually very selective, and it's only going to take serotonin back up into the cell and nothing else. And this is also degraded by MAO. Okay, so monoamine oxidase is going to degrade serotonin as well. So these enzymes, remember that sometimes we have, just like tyrosine, okay, can be a precursor to make things like dopamine and norepinephrine and epinephrine, right? Well, monoamine oxidase, MAO, can degrade several different types of neurotransmitters, okay? So even though we think of neurotransmitters and receptors as being very specific for one another, right? So it's like a lock and a key. Not everything is that way, right? So these degradative, I guess would be the word. Degradative enzymes, okay, can often work at different sites, okay? Serotonin has lots of receptors, so many that we're not even really going to get into it, but we'll talk about a couple of the important ones when we start talking about some of the clinical disorders later on. And there's lots of pathways, and we're going to get to that later too, all right? So again, the big ones, we start off with tryptophan. We have a SERT transporter that brings it back into the cell. It's degraded by MAO. All right. Acetylcholine, one of my favorites. It's one of my favorites because it's one of the oddballs. It's not really an oddball per se, but it is because it's a little bit different in a couple of different ways. Now here, as shown here, and we talked about this already, okay, about how choline, and actually where's choline? Looks like choline's not on here. But anyway, we have, oh, here it is. They have it coming in this way. So choline is combined with acetyl-CoA to actually make acetylcholine. So this is a little bit different than most of the other neurotransmitters. Here you can see it's still getting released, okay? What happens actually with acetylcholine is that we have a very specific enzyme for breaking it down, which is called acetylcholine esterase, all right? That essentially chops it back up into choline and acetate. And the choline gets retaken up into the cell so it can be recycled and used again. All right, this is kind of important because acetylcholine, we don't want it to stick around very long. And one of the reasons we don't want it to stick around very well, or very long rather, is in addition to its prominent role in the central nervous system, acetylcholine is the important neurotransmitter at neuromuscular junctions. So all of the muscles, everywhere where the motor neurons synapse with the muscles in your body, that signaling, okay, that allows those muscles to work is actually based upon the release of acetylcholine. All right, so it's very tightly regulated. We don't want it sticking around very long because you can imagine we don't want your synapses being flooded with neurotransmitter because it could cause all kinds of weird twitching and problems and different types of things, right? So this regulates muscles, as I just said, okay? Autonomic nervous system function. So we talked a little bit about the autonomic nervous system in the beginning. It's also very important in things like learning and memory, okay? There's some really important cholinergic neurons, all right, that are connected with the hippocampal function and things like that. And remember, we talked about the hippocampus as being one of the important brain structures for memory. So acetylcholine is heavily involved in these things as well. And we kind of talked about this a little bit already. So you start out with choline. You have a choline transporter for acetylcholine. So this has also, just like SERT for serotonin, we had the serotonin transporter. Well, here we have the choline transporter that's going to reuptake choline. Remember, it's not acetylcholine because this actually gets chopped in half and then the choline gets reuptaken, all right? And acetylcholine esterase is the one that actually chops that up, all right? Now, what kinds of receptors do we have going on here? Well, acetylcholine binds to different types of receptors. Some of them, well, we'll get into the specific subtypes later, but basically the ones we're going to talk about are nicotinic and muscarinic receptors. So acetylcholine is going to have nicotinic acetylcholine receptors, and it's going to have muscarinic acetylcholine receptors. So two different types. And the last one that I want to go through is norepinephrine, or epinephrine in the synapse, right? So here you can see this diagram basically is showing us all the way from the beginning. So remember, tyrosine is a precursor for all of the amines. So we're going to go L-DOPA, and this time they actually included the L on this one, okay, to dopamine. Now we can have dopamine, we can have the conversion of dopamine to norepinephrine, and then we can actually have the conversion of norepinephrine to epinephrine. Now, this doesn't always happen, right? Because sometimes we stop with dopamine and we have a dopaminergic neuron that's just going to release dopamine, right? Other times we have a adrenergic neuron that might release norepinephrine or epinephrine. Okay, so it just depends. It depends on which one it is. All right. These guys are notorious for being involved in functions like basically anything you can think of that's stress or exercise related. Okay, so in fact, epinephrine is also referred to as adrenaline. A lot of people don't know that. So when you're thinking about getting fired up, maybe you're getting ready to go kickbox somebody or try to kick a soccer ball really far or something like that. I don't know what types of athletic activities or other types of activities you're engaged in outside of the classroom, but you might be getting ready to do something and you're trying to get yourself all pumped up, and you might say, wow, I'm ready to go, my adrenaline's really pumping, right? Well, your adrenaline is actually epinephrine, and your noradrenaline is actually norepinephrine. So these neurotransmitters work on basically everything in you. All of your body tissues respond to these neurotransmitters. Anytime you're exercising or you're scared or you're excited about something, okay, you hear this as the fight or flight sort of response. So giant bear tries to attack you, you go, oh my god, giant bear, I have to run away kind of thing, right? So that's that deal. Also, though, norepinephrine in particular, it's really important for actually focusing, being able to concentrate on tasks and staying alert, okay, and getting aroused from novel sorts of stimuli. Okay, so that's one of the important roles that norepinephrine plays in the central nervous system. Again, tyrosine is the precursor. Now, these norepinephrine is taken up by this norepi transporter. Okay, so the NET, the easy one to remember, NE is usually how we abbreviate norepinephrine, or norepi as I usually call it for short. That's the NET transporter. Okay, not really one for epinephrine so much. Degradation occurs again by MAO and COMT for these guys, all right? And the receptors that these are going to bind to are alpha and beta adrenergic receptors. Now, again, you can remember adrenergic receptor because if you think about it, epinephrine and norepinephrine are really adrenaline and noradrenaline, okay? And in fact, epinephrine is mostly produced by your adrenal gland, your adrenal medulla. Okay, so that's actually in your peripheral system. It's not even really in the CNS. Now there are some neurons that actually do make epinephrine in the brain, but most of the epinephrine is actually produced by your adrenal gland. So these are adrenergic receptors. And there's a lot of overlap between these alpha and beta adrenergic receptors, and both epinephrine and norepinephrine can bind both alpha and beta, and depending on which specific subtype of alpha and beta it is, some of them bind better than others. So they're just little differences, lots of little differences, okay? And again, we're not going to focus on every single one of those small details. We're going to talk about some of the important ones as they relate to certain types of pathology or injury, okay? And we're going to kind of go over things that way in here, all right? So here's a little quiz for you. See how you guys do on it. I hope you enjoyed this installment of neuroscience. The next time that we chat, I believe we will be talking about neural pathways. And there'll be a couple of video clips, and it should be pretty neat. All right? Thanks.